Hello students and my dear friends, myself Madhuvancha. In this session, we'll discuss morphology of bacteria and bacterial cell structure. So these are the learning objectives. After completion of this session, the student should able to define what is prokaryotic cell and list the differences between prokaryotic and eukaryotic cell. Describe the structure of bacterial cell and explain the function of its components. And explain why cell wall forms the basis for classification of bacteria, and explain the structural modification of flagella, like structural modification such as flagella of the cell and their functional importance. So if you see the introduction, as you know, microorganisms, they are living structures of microscopical size. So which means they can be seen under only microscope. So they are the living structures of microscopical size, which means they can be seen under only microscopes. So whenever the microorganisms were first discovered, some of the microorganisms, they are placed under plant kingdom, and some of the microorganisms, they are placed under animal kingdom. So why they have placed under plant and animal kingdom? So some of the microorganisms, they are showing the like branches. So like branching, it is a characteristic of plant. So that's what some of them are placed under plant kingdom. And some of the microorganisms, they are showing the motility, movement. So motility is the characteristic of animal. So that's what they are placed under animal kingdom. But it is unsatisfactory. Why? Because like some of the microorganisms, they are neither branching nor motile. So where to place them? Like lot of confusion. And moreover, like a plant kingdom, like it has the like plants which are visible, and animal kingdom, it deals with animals which are visible, but microorganisms are invisible to the naked eye. So that's what third kingdom established, that is Protista. So here, if you see the Protista, again, it is divided into two groups. The first one is prokaryotes and second one eukaryotes. And if you see prokaryotes, so what is meant by prokaryotes? Here, if you see the word pro means primitive, and karyot means nucleus, which means they have primitive nucleus. Primitive nucleus means there is no nucleolus and there is no nuclear membrane. There is no nucleolus and there is no nuclear membrane. So if you see prokaryotes, they are much smaller and they are measured in the microns. So prokaryotes, they are measured in the microns and they are simple than the eukaryotes. And if you see, they are the molecules, they are surrounded by the membrane and cell wall. They have the cell wall as well as the like cell membrane. And as I told you, like prokaryotes, they have primitive nucleus. Primitive nucleus means there is no nucleolus and no nuclear membrane. There is no true nucleus, and they don't have like membrane bound organelles. Like there is no mitochondria, no endoplasmic reticulum, no Golgi apparatus, no lysozymes, like that. So all these organelles are absent in the prokaryotes. And if you see, what are the examples for prokaryotes? Bacteria and blue-green algae. So bacteria and blue-green algae, so they are the prokaryotes. And if you see bacteria, so bacteria, they are unicellular organisms, which means they are made up of single cells, except higher bacteria, such as actinomycetales. Actinomycetales, it is a higher bacteria, but remaining all bacteria are unicellular, like cocci, bacilli, all they are unicellular. And they do not contain chlorophyll. So if you see bacteria, they do not contain the chlorophyll except BGA, that is blue-green algae. So blue-green algae consists of chlorophyll. And next, eukaryotes. So if you see the word U means true and karyot means nucleus, which means they have true nucleus. So they have, there is clearly defined nucleus. They have like a clearly defined nucleus, which means there is nucleolus, nuclear membrane, and they have well-defined cell structure. They has nuclear membrane and well-defined chromosomes. So like Golgi apparatus, endoplasmic reticulum, mitochondria, all these organelles are present in the eukaryotes. And if you see, what are the examples for eukaryotes? Like fungi, protozoa, slim molds, they comes under eukaryotes. And see, these are the differences between prokaryotes and eukaryote cells. If you see nucleus, the character nucleus, nuclear membrane is absent in the prokaryotes, and nuclear membrane is present in the eukaryotes. And nucleolus is absent in the prokaryotes. That means it is in bacteria. So nucleolus is absent in the bacteria and it is present in the eukaryotes. And chromosome 1, that is circular chromosome, is present. And here in eukaryotes, 1 or more paired and linear. And if you see cell division, so the prokaryotes, it divides, the bacteria, it divides by the process called binary fission. And eukaryotes, they divide by the process called mitosis. And next, cytoplasmic membrane, structure and composition. So fluid phospholipid bilayer, but they do not possess the sterols. They lack the sterols. And here eukaryotes also fluid phospholipid bilayer, but they consist of sterols. So here sterols are absent in the bacteria, except in mycoplasma. So mycoplasma, it is a bacteria. So only mycoplasma possess the sterols. So remaining bacteria, they do not possess the sterols. And next, cytoplasm. So cytoplasmic organelles like mitochondria, lysosomes, Golgi apparatus, endoplasmic reticulum, vacuoles, all these organelles are absent in the prokaryotes. That means bacteria. So all these organelles are absent in the bacteria, and all these organelles are present in the eukaryotes. And ribosomes are present both in prokaryotes and eukaryotes. In bacteria, 70 S type of ribosomes are present. Bacteria consist of 70 S type of ribosomes, and eukaryotes, they consist of 80 S type of ribosomes. So S stands for Swedberg unit. Next, cell wall. In prokaryotes, so cell wall is present. In bacteria, cell wall is present, and the cell wall, it is made up of peptidoglycan. Bacterial cell wall is made up of peptidoglycan, which is a complex carbohydrate. And in case of eukaryotes, in case of animals and protozoans, the cell wall is absent. And in case of plants, fungi, algae, cell wall is present. And here eukaryote cell wall is made up of cellulose. If you see plants and others, their cell wall is made up of cellulose, and fungal cell wall is made up of chitin. Locomotor organelles. What are locomotor organelles? Locomotor organelles which helpful for motility. They helpful for the movement of the organism. So prokaryotes, bacteria, they are motile with the help of the flagella. So flagella is the organ of the locomotion. So eukaryotes, they have either flagella or cilia. Next, size of bacteria. So here, the unit of measurement, the bacterial size, it is measured in micron or micrometer. Bacterial size is measured in micron or micrometer. So what is 1 micron or 1 micrometer? So 1 micrometer is equal to 1 by thousands of the millimeter. And viral size is measured in the nanometers. So viral size is measured in the nanometer. So what is 1 nanometer? That is 1 by thousands of the micrometer. Here, nanometer is smaller than the micrometer, which means viruses are smaller than the micrometer. If you see in generally, bacteria and fungi, they can be observed under compound microscopes, okay, which are measured in micron or micrometer. So whereas viruses, like as they are smaller than the bacteria, they are not visualized under compound microscope. So we need electron microscopy. And bacteria of medically important, like the bacteria of medical importance, like E. coli or staphylococci or any other bacteria. So this is general size, that is having 0.2 to 1.5 micrometer in the diameter and 2 to 5 micrometer in the length. Next, morphology of bacteria. So based upon the shape and based upon the other characteristics, so bacteria, they are mainly classified into 6 types. So based upon the morphology or based upon the shape, the bacteria, they are classified mainly into 6 types. So the first one is cocci, and second one bacilli, Third one, spirochetes. Fourth one, actinomycetes. Fifth one, mycoplasmas. Sixth one, rickettsia and chlamydia. If you see the cocci, so what is cocci? So cocos means berries. The word cocos means berries. They are oval shaped or spherical shaped bacteria. They are oval shaped or spherical shaped bacteria, which means they are round or oval in the shape. Next, the word bacillus. The word bacillus means rod. So bacillus, it is a rod-shaped bacteria. They are rod-shaped cells. And next, spirochetes. So spiro means coil. The word spira means coil and cheate means hair-like. So spirochetes are long, slender, flexible, spiral forms. So spira means coil, cheate means hair-like. So like they are like slender, flexible, spiral forms. So like treponema. So the example is like treponema. And next, actinomycetes. So the word actis means ray. The word actis means ray and mycus means fungus. So they are branching filamentous bacteria. They show the branches. So it resembles like fungal mycelium. It resembles a fungal mycelium, but they have rigid cell wall. And next, mycoplasmas. Mycoplasmas, they are cell wall deficient bacteria. They lack the cell wall. So they do not possess the cell wall. So as they do not possess the cell wall, so they do not possess a stable shape as they have no cell wall. So generally, cell wall only gives shape and rigidity to the bacteria. So as there is no cell wall, so there is no like a proper shape or there is no stable shape. They occur as round or oval bodies or interlacing filaments. So their size is also very small. So generally in between 50 to 300 nanometer in the diameter, and they can reproduce in cell-free medium. Next, rickettsia and chlamydia. So they are small, obligate intracellular bacteria. They are obligate intracellular bacteria. So the word obligate means strict. So strictly, they live inside of the cells only. They live inside of the host cells. So actually, obligate intracellular parasitism, it is a characteristic of viruses. So generally, viruses are also obligate intracellular infectious agents. So these rickettsia and chlamydia, they were previously considered as viruses, but due to their typical cell structure, they have the cell structure and like they are susceptible to the antibiotics and they have bacterial enzymes and they have structural similarities. So because of all these characteristics, again, they are reclassified as the bacteria. And next, arrangement of bacteria. So we'll see the arrangement of the cocci. So as we discussed in the previous slide, cocci or coccus means what is the shape of the cocci? That is oval or spherical in the shape. In the picture you can see. And next, if two cocci are arranged as pair, that is called as diplococcus. So diplo means two. If two cocci are arranged as a pair, so that is called as diplococcus. So generally, pneumococcus, meningococcus, they are arranged as diplococci. So that is two cocci are arranged as a pair. And next, if cocci, if they are arranged in the chain form, that is called as streptococci. So the word strepto means a chain. So if the cocci, if they are arranged in the chain form, that is called as streptococci. Next, staphylococci. So what is meant by stapule? So stapule means as a bunches of grapes or as a cluster. The word stapule means as a bunches of grapes or as a cluster. So if the cocci, if they are arranged as a bunches of grapes or as a cluster, then it is staphylococci. Next, tetrad, which means group of four. Okay, four cocci are arranged as a group. And next, sarcina, that is a group of 8. So 8 cocci are arranged as a group. Next, arrangement of bacilli. So as we discussed in the previous slide, so bacilli, it is a rod-shaped bacteria. So in the bacilli, like various forms are there, like Coccobacillus. So what is Coccobacillus? The length of the bacteria is approximately same as width. The length of the bacteria is approximately same as the width. So that is Cocobacillus. So example, Brucella. Brucella species, like they are the Cocobacillus. Next, Diplobacilli. So Diplobacilli means so two bacilli are arranged as a pair. So example, Moraxilla lacunata. Moraxilla lacunata, it's a Diplobacilli. Next, Streptobacilli. So strepto means chain. So the bacilli, they are arranged in the chain form. Bacilli are arranged in the chain form. This is the Streptobacilli. Next, palisades arrangement. So what is palisades arrangement? So one edge of the bacilli or one end of the bacilli is attached to the another edge or another end of the bacilli. So this is palisades arrangement. So here diphterioids, they are arranged as palisades. Next, another forms of the bacilli, other forms of the bacilli like comma shaped. So they are curved appearance. They appears like comma. Example like a Vibrio color. Next, as we discussed, spirochetes. So what are the spirochetes? So they are long, thin, flexible spiral forms. And next, spirilla. So spirilla, they are short, rigid spiral forms. So spirochetes are long, thin, flexible spiral forms. Spirilla, they are short, rigid spiral forms. And next, this is Chinese letter arrangement or cuneiform arrangement. See this Corynebacterium diphtheriae. So they arrange as Chinese letter pattern or cuneiform arrangement. Next, anatomy of a bacterial cell or structure of bacterial cell. So in the picture, you can see this is the structure of bacterial cell. So bacterial cell structure is very simple. So when compared to the eukaryotic organisms, so this Golgi apparatus, endoplasmic reticulum, mitochondria, lysosomes, all these organelles are absent. So here in the picture, you can see they mainly possess the a cell wall. So cell wall, this is the outermost layer of the bacteria, and beneath the cell wall, a plasma membrane is present, and these are fimbriae or pili. Next, flagella, which is the organ of locomotion or which helpful for motility. So fimbriae or pili, they helpful for attachment. They are the organs of adhesion. And next, plasmids. So plasmids are the extra chromosomal genetic elements which consist of DNA. And this is nuclear area or nucleoid, which consists of DNA. And you can see the plasma membrane and cell wall. And next, ribosomes. So ribosomes, they are the centers of protein synthesis. And this is cytoplasm. And this layer is capsule. So don't get confused as capsule. So as outer layer. So capsule is not a outer layer because very few bacteria possess the capsule. So that's why we are not supposed to consider capsule as outer layer. So we should consider cell wall as outer layer. Next, mesosomes. So mesosomes, they are the principal sites of the respiratory enzymes. And next, inclusion bodies. So they store the energy. So this is very simple. So we'll discuss in detail one by one. So here, the bacterial cell structure, it is divided into three categories like outer layer. So outer layer, again, it consists of two components. The first one is rigid cell wall and second one cytoplasmic membrane, or it is also called as cell membrane or plasma membrane. So this cytoplasmic membrane, it present beneath the cell wall. And next, cytoplasm. So cytoplasm includes cytoplasmic inclusions, ribosomes, mesosomes, and nucleus. And next, additional structures. Additional structures are plasmid, slime layer, capsule, flagella, fimbriae or pili, and spores. So these are the additional structures. So we'll start with the cell wall. So cell wall is the outermost layer. Cell wall is outermost layer. It is a tough and rigid structure. It is a tough and rigid structure. It encloses the cytoplasm and it gives shape and rigidity to the bacteria. And it is 10 to 25 nanometer in the thickness and 20 to 30% of dry weight of the cell. And it takes part in the cell division. So if you see the cell wall, it is made up of complex polysaccharides, that is peptidoglycan, or which is mucopeptide. So this peptidoglycan, again, it consists of N-acetyl glucosamine and N-acetyl muramic acid, NAM, NAG, NAM, NAG. Okay. So this is a structure of peptidoglycan. This peptidoglycan, it consists of N-acetyl glucosamine and N-acetyl muramic acid alternating in the chains NAM, NAG, NAM, NAG, and they are held together by the tetrapeptide chains. So these NAM, they are held together by like a peptide bonds or like a peptide chains. This cell wall, it carries the bacterial antigens. So this, as this bacteria, cell wall act as antigens, so it plays important role in the virulence. What is virulence? It is the ability of a strain to produce the infection is called as the like virulence. So it increases the virulence of the bacteria, and it also like contribute for the like immunity. Why? Because as it acts as antigens, so the specific antibodies are formed. So these antibodies indirectly they give the immunity. Based upon the nature of cell wall, Hans Christian Gram, he divided bacteria mainly into two groups, gram-positive bacteria and gram-negative bacteria. So after gram staining, gram-positive bacteria appears purple or violet in the color. In the picture you can see gram-positive bacteria appears purple or violet in the color, and gram-negative bacteria appears pink or red in the color. So here, this is gram-positive cocci, which is spherical or oval in the shape. They are violet in the color. And this is gram-positive bacilli, which are rod-shaped bacteria. They also appears purple or violet in the color. And in the picture, you can see gram-negative bacilli. So this gram-negative bacilli and gram-negative cocci, so they appears pink or red in the color. Then what is gram-positive and what is gram-negative? So what is the difference between gram-positive and gram-negative? So here, gram-positive bacteria has simple chemical nature than the gram-negative bacteria. So we'll see in detail about gram-positive cell wall and gram-negative cell wall. So, so this is cell wall. It is a target for many of the antibiotics. So this is several antibiotics. They interfere with the like cell wall synthesis, such as penicillin and cephalosporin. If you see these penicillins and cephalosporins, they act upon the peptidoglycan of the cell wall. They act upon the peptidoglycan of the cell wall. So these penicillins, they damages the peptidoglycan of cell wall. So if the peptidoglycan of cell wall is damaged, automatically bacteria cell is damaged. This is the structure of gram-positive cell wall. So if you see, this is the cell wall, and next beneath the like cell wall, cytoplasmic membrane is present. And if you see, the peptidoglycan content is more in the gram-positive. So gram-positive cell wall, it consists of thick multi-layered peptidoglycan. So it is present like it consists of thick multi-layered peptidoglycan, and so generally it is thicker, around like 16 to 18 nanometer. Whereas gram-negative cell wall consists of less peptidoglycan, and they have teichoic acids. So these teichoic acids generally, like they are the major surface antigens of gram-positive bacteria, and they are water-soluble polymers. They contains ribitol or glycerol polymers. And see these teichoic acids, there are two types: cell wall teichoic acid and membrane teichoic acid. The cell wall teichoic acid is covalently linked to the peptidoglycan, and membrane teichoic acid to the cytoplasmic membrane. And next, they have like lipoteichoic acids. So these lipoteichoic acids, they are extended to the cytoplasmic membrane. And these teichoic acids, they are embedded in the peptidoglycan, and these lipoteichoic acids, they are extended to the cytoplasmic membrane. Next, gram-negative cell wall. So in the gram-negative cell wall, the peptidoglycan content is less. The peptidoglycan content is less and it is complex structure. So it consists of outer membrane and like a peptidoglycan and a cytoplasmic membrane. So here the peptidoglycan content is less. So it consists of single layered peptidoglycan and there is a space between outer membrane and cytoplasmic membrane. So this space is called as periplasmic space. So this peptidoglycan, it is present in between the like a periplasmic space, and this outer membrane includes porin, and so these porins, they allow the like a passage of small hydrophilic molecules across the like a membrane, and the lipopolysaccharides, they are extended to the extracellular space. So these are the differences between gram-positive and gram-negative bacteria. If you see the property of bacteria and gram-positive and gram-negative, thickness of wall, it is 20 to 80 nanometer, and it is 10 nanometer. So number of layers which are present in the cell wall, in gram-positive only one layer is present, that is peptidoglycan layer. Whereas in case of gram-negative bacteria, two layers are present. One is outer membrane and another is the peptidoglycan. And here peptidoglycan content is more in gram-positive, that is more than 50% of like a gram-positive cell wall consists of peptidoglycan. And in case of gram-negative, the peptidoglycan content is less, that is 10 to 20% only. And the teichoic acids, they are present in the gram-positive, and the teichoic acids are absent in the gram-negative. And lipoprotein content is less in case of gram-positive, that is in between 0 to 3% only. And in case of gram-negative, the lipid and the lipoprotein content is more, that is 58%. And protein content is 0. And here, in case of gram-negative bacteria, that is 9%. And the lipopolysaccharides, they are 0. And here, in the gram-negative, 13%. So sensitive to the penicillin. Yes, gram-positive bacteria is sensitive to the penicillin. Why? Because like penicillin, they mainly act upon the peptidoglycan of cell wall. They mainly damages the peptidoglycan of cell wall. So as gram-positive cell wall has more peptidoglycan, so this gram-positive bacteria is sensitive to the penicillin, and gram-negative bacteria is less sensitive to the penicillin. Next, digested by the lysosomes. Gram-positive, yes, it is easily digested by the lysosome, and gram-negative, it is weakly digested by the lysosomes. So demonstration of cell wall. So how like we can see the cell wall. So generally the cell wall cannot be seen by the light microscope, and they does not stain with the simple dyes. So how we can see the like a cell wall, how we can demonstrate the cell wall? The first one is plasmolysis. So what is plasmolysis? So when the bacteria is placed in hypertonic saline. So in the picture you can see whenever the bacteria is placed in the hypertonic saline, so we can see the sudden shrinkage of the cytoplasm occurs. So here the cell wall, it retains its original shape and size. So there is no change in the cell wall shape and size, but there is sudden shrinkage of the like cytoplasm. This is called as plasmolysis, and it can be demonstrated by microdissection, differential staining, and as it acts as antigen, it reacts with its specific antibody, and we can observe under electron microscopy also. Next, bacteria with defective cell wall. So the synthesis of cell wall, it is inhibited or interfered by different factors. So what are those factors? Like antibiotics, bacteriophages, and lysosome. So lysosome, that is normally present in the tissue fluids. So what this lysosome to do? It lyses the susceptible bacteria by splitting the linkage of peptidoglycan in the cell wall. So this lysosome, it mainly act upon the peptidoglycan of the cell wall. It splits the like a peptidoglycan of the cell wall. So mostly lysosomes, as I told you, they act upon the gram-positive bacterium in a hypertonic solution so that the protoplast is formed. And in case of gram-negative bacteria, the spiroplast is formed. So generally, these bacteria without the cell wall or bacteria with a defective cell wall, they place in the role persistent chronic infections, such as in case of pyelonephritis. So they plays a major role in case of pyelonephritis infections. Bacteria without the cell walls, they are of mainly four types. First one is mycoplasma, second one L forms, third one protoplast, fourth one spiroplast. So if you see mycoplasmas, so what is mycoplasma? Generally, they are naturally occurring bacteria without the cell walls. So they are classified as separate genus. They do not require hypertonic environment for its maintenance, and they are stable in culture medium. And next, L forms, Kleiniberger Nobel, while he was studying about like Streptobacillus forms in the Lister Institute, London, he observed some abnormal forms of the bacteria, and he named them as L forms to honor the like Lister Institute, after the like Lister Institute. So how these L forms they develop? They develop either spontaneously or in the presence of penicillin or in the presence of other agents. So they develop like in the presence of penicillin or other agents. And what this penicillin or other agents they do, they interfere with the synthesis of cell walls. These L forms are difficult to cultivate, and they require agar containing solid medium, which is having the like right osmotic strength. And L forms, they are sometimes spontaneously formed in patients treated with the penicillin. They are more stable than the protoplast and spiroplast. If you see protoplast, as we discussed, protoplast, they are derived mainly from the gram-positive bacteria. They contain cytoplasmic membrane and the cell wall is totally absent. And they are produced artificially by lysozyme in hypertonic medium. They are unstable. So hypertonic condition is necessary for maintenance. Next is spiroplast. They are derived from the gram-negative. They are produced in the presence of penicillin, and they are osmotically fragile, and they must be maintained in the hypertonic culture medium. Next, cytoplasmic membrane or plasma membrane, or it is also called as cell membrane. So here, the cytoplasmic membrane, it is a thin layer which is having the size of 5 to 10 nanometer. It is having the size of 5 to 10 nanometer. And cytoplasmic membrane, it present beneath the cell wall. So it separates the cell wall from the cytoplasm. And when we observe under electron microscope, it shows trilaminar structure. So trilaminar structure means three layers, they are present as a unit membrane. They constitute a unit membrane. They act as the semi-permeable membrane. So what is semi-permeable membrane? It controls the inflow and outflow of metabolites. Semi-permeable membrane, it controls the inflow and outflow of metabolites. It is made up of lipoproteins, and they contain the small amount of carbohydrates. So generally, sterols are absent in the bacteria, except in mycoplasma. So as we discussed, mycoplasma, they are cell wall deficient bacteria. So as there is no cell wall, so they have the cytoplasmic membrane, which is consists of sterols. And this cytoplasmic membrane, it acts as osmotic barrier, and they consist of numerous enzymes such as permease, oxidase, polymerase. So what these enzymes they do? They involve in the active transport of the selective nutrients, and they contain cytochrome oxidase, and it consists of enzymes of tricarboxylic acid cycle and enzymes of necessary for the cell wall synthesis. It consists of enzymes which is necessary for cell wall synthesis. Next, cytoplasm. What is cytoplasm? It is a colloidal system of various organic and inorganic solutes in a viscous water solution. So it differs from eukaryotic cytoplasm. How it differs from eukaryotic cytoplasm? Because they do not possess mitochondria, endoplasmic reticulum. So these mitochondria and endoplasmic reticulum, they are present in eukaryotes and they are absent in the bacteria. They contain water, ribosomes, mesosomes, vacuoles, and inclusions. And they stain with the basic dyes. They stains uniformly with the like basic dyes in the young cultures. Next, ribosomes. So here in this picture, you can see these are the like ribosomes. Ribosomes, they are the centers of protein synthesis. They are made up of rRNA and ribosomal proteins. They are made up of ribosomal RNA and ribosomal proteins. They have the size of 10 to 20 nanometer. They are 10 to 20 nanometer in the size, and generally 70 S type of ribosomes are present in the bacteria. And again, 70S, it has two subunits, that is 30S subunit and 50S subunit. S stands for Swedberg unit. Next, intracytoplasmic inclusions. So in the picture, you can see these are intracytoplasmic inclusions. They are the sources of stored energy. So they store the energy and they're present in only in some species of bacteria. We don't know the exact function of intracytoplasmic inclusions. So they are frequent in the bacteria which are grown under nutritional deficiency. Example, metachromatic granules in diphtheria bacilli. So they are present as like polymetaphosphate, lipid, polysaccharide, and granules of sulfur. So as I told you, they are more frequent in the bacteria which is grown under nutritional deficiency. And whenever the nutritional deficiency is corrected, so whenever the nutrients are available, so these inclusion bodies they disappear. And this inclusion bodies, they also helpful in the diagnosis of diphtheria infection. Metachromatic granules, they helpful in diagnosis of diphtheria bacilli. Next, mesosomes. So mesosomes, they are multilaminated structures. They are formed by invaginations of plasma membrane. So in the picture, you can see this layer is the cytoplasmic membrane. So mesosomes, they are formed by invaginations of plasma membrane. They are the principal sites of respiratory enzymes. So they are analogous to mitochondria of eukaryotes. So in case of eukaryotes, mitochondria helpful for respiration. So here as mitochondria is absent, so mesosomes they act as sites of the respiratory enzymes. They coordinate nuclear and cytoplasmic division during the binary fission. They are more prominent in the gram-positive bacteria. There are two types of mesosomes are there. One is septal and another is lateral. Next, nucleoid. So bacteria, they do not have the true nucleus. So as you know, they do not have the true nucleus. There is no nucleolus and there is no nuclear membrane. The genome is single, circular, double-stranded DNA it is present. The DNA is haploid and it divides by binary fission. And this DNA, it cannot be demonstrated by ordinary microscope. DNA cannot be demonstrated by ordinary microscope, and it measures 1 mm when straightened. Next, plasmid. So what are the plasmids? Plasmids are the extra chromosomal genetic elements which consist of DNA. So they are not essential for the life of the cells. So without the plasmids also, these bacteria can survive. And plasmids are transmitted from one a mother cell to the daughter cells during the binary fission. So bacteria divides by process called binary fission. So during binary fissions also, the plasmids are transferred from a mother cell to the daughter cell. And they are also transferred from one bacterium to the another bacterium by the process called as conjugation. So what is the conjugation? It is a process of transfer of DNA from F plus cell to the F minus cell. So during the conjugation process, so these plasmids, they can they transmit from F plus cell to the F minus cell, and they confer certain properties such as they give the drug resistance and toxicity. They are responsible for drug resistance and toxicity. Next, bacterial capsule and slime layer. So capsule, it is the viscous layer, amorphous viscous layer, which is secreted around the cell wall. In the picture you can see, so this is the capsule. So this is the viscous layer which is secreted around the cell wall. So generally the capsule, it is polysaccharide or polypeptide in the nature. So all bacterial capsule, it is polysaccharide in the nature, but Bacillus anthracis capsule is polypeptide in the nature. So what is capsule? So capsule, it is sharply defined structure and it is antigenic in the nature. So example, in case of Streptococcus pneumoniae. So when this secretion is organized into the like a sharply defined structure, so it is known as the like a capsule. Suppose if the capsule, if it is very thin and if it cannot be demonstrated under normal microscope, if it cannot be seen under normal microscope, then it is called as microcapsule. So here microcapsule, it is present in Neisseria meningitidis. So as I told you, like the capsule, it is polysaccharide or polypeptide in the nature. So Bacillus anthracis capsule is polypeptide in the nature. Then what is a slim layer? So that is the like a loose undemarcated secretions. So whenever this amorphous, so this like a capsule, it is like a loose undemarcated secretion. Next, functions of capsule. So what are the functions of capsule? So this capsule enhances the bacterial virulence by inhibiting the phagocytosis. So what is phagocytosis? Phagocytosis, it is a engulfment of bacteria by the like neutrophils. So here, as this capsule present as a like a protective covering. So as it is present like above the like cell wall, so it acts as like a protective covering, and it like so as I told you like in the previous slide, so some of the antibiotics such as penicillin, so penicillin, they act upon the like a cell wall of the bacteria. So as this capsule, it is present as protective covering above the like a cell wall, so generally these antibacterial agents, they cannot work on the like cell wall, so it acts as protective covering, and they increases the like virulence. So with the virulence, it is the ability of a strain to produce the like disease. So it inhibits the phagocytosis process, which is a protective mechanism. And here this capsular antigen, it is specific for each type of bacteria. So this capsule, it is helpful for identification and typing of bacteria. So these are the examples for capsulated organisms. Example, first one, Streptococcus pneumoniae, a Klebsiella species, Bacillus anthracis, Haemophilus influenza, Cryptococcus neoformans. So the first four are bacteria, and the last one, that is Cryptococcus neoformans, it is a fungus. Demonstration of capsule. So how we can identify the capsule? So here, like we can identify it by the India ink staining, or it is also called as negative staining. Next, serological method such as capsule swelling reaction or Quelling reaction. So in the India ink staining or in the negative staining, so the like a bacterial culture, it is mixed with either India ink or nigrosin on the slide, and a smear it is prepared and apply the like a cover slip. So here the background gets a stain and the organism also stain. But here the capsule, they do not take up any stain. They do not absorb any color. So that's what this capsule appears as clear halo structure around the bacterium. Capsule appears as clear halo structure around the bacterium. Next, serological methods such as capsule swelling reaction or Quelling reaction. So in this, a suspension of bacterial, like a capsulated bacterium, it is mixed with anti-capsular serum. So this is before addition of antiserum, and this is after addition of antiserum. Here capsulated bacterium, it is mixed with a specific anti-capsular serum, and it is examined under microscope. So in the microscope, we can see the like capsules are swollen. So under microscope, we can see the like a swollen capsule. It is due to the like increase in its refractivity. So this phenomenon is called as capsule swelling or Quelling reaction. Next, clinical significance such as capsular antigen. So as we discussed, this capsule acts as antigen. So this antigen can be detected in the specimen like CSF. It can be detected by latex agglutination test by using of latex particles which are coated with a specific anti-capsular antibodies. So examples like latex agglutination test, they are available for detection of Streptococcus pneumoniae and Cryptococcus neoformans, which is a fungal infection. Next, McFadden capsule stain. So Bacillus anthracis capsule. So the capsule of Bacillus anthracis, it is demonstrated by using of polychrome methylene blue stain. Here it is stained with the polychrome methylene blue. Next, vaccine. So here, like the capsules of some of the bacteria, they act as antigen. So as they act as antigen, like anti-capsular antibodies are formed, which are protective in the nature. So these capsular organisms used as the vaccines, like meningococcal vaccine, pneumococcal vaccine, and Haemophilus influenza type B vaccine. Next, flagella. So flagella, they are long, which are having the size of 5 to 20 micrometer in the length and 0.01 to 0.02 micrometer in the diameter. And they are filamentous cytoplasmic appendages protruding through the cell wall. They are the surface appendages. They are cytoplasmic appendages protrude through the cell wall. They are the organs of locomotion, which helpful for motility. They are made up of proteins, and that proteins are called as flagellants. Flagella is made up of protein, and those proteins are called as flagellants. So the number and distribution of flagella on the bacterial surface, it is characteristic for each species. So based upon the number of flagella and based upon the distribution of flagella, we can classify the bacteria. And how we can see the like presence of flagella, or how we can observe the like motility. So with the help of hanging drop preparation, we can see the motility of the bacteria. So all motile bacteria, they possess one or more flagella except spirochetes. Suppose if you see in case of Treponema, they have special characteristics. They have endoflagella. The flagellate is present inside of the bacteria. If you see the parts and composition, each flagellum, it consists of three parts. The first one is filament, and second one is hook, and the third one is basal body. And if you see the filament, it lies to the it is lies to the external to the cell, and it is connected to the hook at the cell surface. So filament lies external to the cell, and it is connected to the hook at the cell surface. And the second one is hook. Hook, it is like a basal body portion is embedded in the like a cell envelope. And next, basal body. So basal body consists of outer and inner rings. It consists of outer rings and inner rings. So these are attached to the like a cytoplasmic membrane. So these outer rings are absent in the gram-positive bacteria. Next, types of flagellar arrangement. So here, based upon the flagellar arrangement, so we can classify the bacteria like monotrichous, lophotrichous, amphitrichous, and peritrichous. So if you see polar or monotrichous, which means one flagella present at one pole or one flagella present at one end. So single flagella present at one end. So example in case of Vibrio cholera, and in case of Pseudomonas, we can see like only one flagella. Next, lophotrichous. So lophotrichous means tuft flagella present at one end or tuft flagella present at one pole. Example, spirillum. Next, amphitrichous. So amphi means both. So here flagella present at both the ends or flagella present at both the poles. Example, alkali genus fecalis. Next, peritrichous. Peri means all over the cell. So here peritrichous means flagella, it is present all over the cell. So example in case of Salmonella typhi. Next, fimbriae or pili. They are thin, hair-like appendages. They are present on the surface of many gram-negative bacteria. They have the size of 0.1 to 1.0 micrometer in the length and less than 10 nanometer in the thickness. They are shorter and they are thinner than the flagella. And they are organs of adhesion. They are helpful for the attachment. They allow the bacteria to colonize on environmental surfaces are cells and they resist to flushing. They strongly attaches to the host body and they are made up of proteins and that proteins are called as pillins. Almost like 100 to 500 fimbriae present peritrachusly. All over the cell, the fimbriae is present. So they develop in the freshly isolated strains and in the liquid cultures, but they disappear in subcultures on the solid media. So if you go for like subcultures on solid media, so these fimbriae, it is disappeared. And fimbriae, it is not at all related to the motility. So they do not contribute for the motility. Just they are the organs of adhesion. They are present both in motile as well as non-motile bacteria. And next types and functions of pili. So pili, it is of three types. The first one is common pili, and second one sex pili, and third one colon pili or colicin pili. So if you see common pili, they are short and abundant in the number. They are adhesive and antigenic. They are helpful for attachment and they act as antigenic determinant. And next, sex pili. So sex pili helps in the conjugation. So in the picture, we can see like what is conjugation? It is a process of transfer of DNA from F plus cell to the F minus cell. That is from donor cell to the like recipient cell. So here, with the help of this pili only, this F plus cell attaches to the F minus cell and it becomes the like a conjugation tube. So it helpful for transfer of the like genetic material. And next, colon or colicin pili. Detection of fimbriae. So how we can detect the fimbriae? So fimbriae can be detected by electron microscopy and hemagglutination. So what is hemagglutination? So these like fimbriated bacteria, they agglutinate with RBC of guinea pigs, fowl, horses, and pigs, human and sheep cells also, but weakly. This property of hemagglutination, it is a simple method for detecting the presence of the fimbriae. Next, bacterial spores. So spores are highly resistant stages. They are formed under unfavorable conditions. So what are unfavorable conditions? Like if there is no suitable environment, if nutrients are not available, if the temperature is not suitable, and if the pH of culture medium is changed. So there are many factors are there. So all these are like unfavorable conditions. So whenever the bacteria has unfavorable conditions, so they turn into the like a spore. And these spores, they are formed inside of the bacterial body. That's why they are called as the endospores. And sporulation, it is not a method of reproduction. In each cell, only one spore is formed. So next, sporulation. So what is the sporulation? So sporulation means a process of spore formation. Process of spore formation, it is called as the like sporulation. So here the spores, they develops from a portion of protoplasm near the one end of the cell. It develops from a near end of like near to the one end of the cell, and the remaining part of cell is called as the like sporangium. So first of all, the DNA it replicates and it divides into two. One is incorporated into the sporangium and another is incorporated into the forespore. And a transverse septum, it grows the like it grows across the cell from the like cell membrane, and it divides the forespore and the sporangium. It divides forespore and the sporangium. And see, this forespore, it is completely encircled by the septum as a double layer membrane. And inner layer become the spore membrane. Inner layer become the spore membrane and outer become the spore coat. So between these two layers, so this is outer membrane and this is inner membrane. So between these two layers, the spore cortex is formed. So next, shape and position of spore. So here, the spores, they may be central or subterminal or terminal. And the shape of the spores may be either oval or spherical. And the width of the spores, so the diameter of the spore is same or less than the like width of the bacteria. So like non-bulging means they do not come out from the like a bacterial body. So here bulging spores. So bulging spores means that they are wider than the like a bacteria. So here in this slide we can see like spores. There are mainly three types, like non-bulging spores, bulging spores, and free spores. So here this is the shape of the spore is oval and it is present in the center of the bacteria. This is oval central. And here the shape of the like spore is spherical and it is present in the center. And here the shape of the spore is oval and they're present in the subterminal. So this is oval subterminal. So these are non-bulging spores. And here bulging spores means, so here we can see the like they come out from the like a little bit bacterial body. So here like the shape of the spore is oval and it is subterminal. And here it is oval terminal and this is spherical terminal and it is a free spore. Next, resistance. See, the spores are highly resistant to the ordinary boiling. With the ordinary boiling or by normal disinfectants and with the normal heating, we can't destroy the like spores. They are resistant structures, so they cannot be killed by using of ordinary boiling or disinfectants or heat. So why they are that much resistant? Why? Because they have high content of calcium and diplonic acid and they have low water content. So because of that one, they are highly resistant. Then how we can kill the like spores? We can kill the like spores like by autoclaving at 121 degrees centigrade, 15 lb pressure for 15 minutes. And some of the disinfection procedures also they kill the like spores. Next, germination. So what is meant by germination? It is a process of conversion of spore into vegetative form is called as the germination. So whenever the bacteria has favorable conditions, like whenever the bacteria has nutrients, or if the temperature is suitable, or if the pH is suitable, if like environmental conditions are suitable, then the spore, it will turn into the like a vegetative form. That is called as the germination. Demonstration. How we can identify the like spores. So gram staining. In the gram staining, the spore appears as unstained retractile body. They do not take up any stain. And it is also identified by modified Gill-Nielsen staining. So in the modified Gill-Nielsen staining, the spores appears red in the color. The spores appears as acid first, that is red in the color. So generally, we use like sulfuric acid for decolorization in the Jaden staining. But in case of modified Gill-Nielsen staining, especially for spores, only 0.25 to 0.5% of sulfuric acid is used. So instead of 20% sulfuric acid, we use 0.25 to 0.5% of sulfuric acid is used. Uses of spores. So what are the uses of spores? So they are indicator for proper sterilization. Example like a Geobacillus stereothermophilus. So it is destroyed at a temperature of 121 degrees centigrade for 10 to 20 minutes. So that is used in the temperature of autoclaves. So these spores, they are kept inside of the autoclave before sterilization. So after sterilization, if the absence of spores, so if there are no spores, that indicates that the sterilization process is successful. That sterilization is done, happened properly. And the spores of Bacillus atrophiceus, they are used as a sterilization control for ethylene oxide sterilizer. So next to spore forming bacteria. So examples for spore forming bacteria: obligate aerobes. Aerobes means which grows in the presence of oxygen. Strict aerobes Bacillus anthracis and Bacillus subtilis. So these are spore forming bacteria. Next, obligate anaerobes, which are strict anaerobes. Strictly, they grow under absence of oxygen, such as Clostridium tetani, Clostridium welchii, or Clostridium perfringens, and Clostridium botulinum. So these are anaerobes. These are like a spore-forming bacteria. And if you see, both Bacillus and Clostridia, they are gram-positive bacilli. Next, pleomorphism and involution forms. So what is mean by pleomorphism and what is mean by involution forms? So pleomorphism means a great variation in shape and size of individual cells. So they show the like a variation in the shape and size of the cells, that is called as like a pleomorphism. Example, Proteus species or existing in more than one form is also called as a pleomorphism. So Proteus bacteria, it has no proper shape and proper size. Next, involution forms. So what are involution forms? They are the like swollen and aberrant forms in the aging cultures. So especially in case of high salt concentration, we can see the like swollen and aberrant forms. So example, plague bacillus, that is Yersinia pestis. This is the end of the session. So if you see the like summary, bacteria exhibit many shapes such as bacilli, foci, spirochetes, and actinomycetes. And the bacterial size is measured in microns. Bacterial cell, it consists of different components like cell wall, cytoplasmic membrane, cytoplasm, nucleus, capsule, flagella, fimbriae, and bacterial spores. Thank you.